This video is about SERCA, sarcoendoplasmic reticulum calcium ATPase. It is a protein or enzyme that uses ATP to pump substrate against their gradient. Especially this SERCA uses calcium as a substrate to transport it inside the sarcoplasmic reticulum or endoplasmic reticulum with the help of ATP, and it pumps out three proton outside the cell. So SERCA stands for sarcoendoplasmic reticulum ATPase, calcium ATPase. If we make a rough diagram of this protein, just to get to know the function of this protein, let's assume this is a SERCA. Then what it does is it pumps out three proton for two calcium ion by hydrolyzing ATP into ADP and inorganic phosphate. This occurs mainly on the and on the membrane of endoplasmic reticulum or sarcoplasmic reticulum, depending on which cell it is. If it is a muscle cell, then we will call it sarcoplasmic reticulum. If it is a normal cell, then we are bound to call it endoplasmic reticulum. Now let's discuss about the family the SERCA belongs to. Actually, ATPases have different kinds of members in it. First one is FATPase, second one is VATPase, third is AATPase, fourth is PATPase, and now fifth is EATPase. SERCA belongs to PATPase. Let's get a gist of about what these ATPases are about. FATPases are used for formation of ATP by using proton gradient. These are present in mitochondria, on the cristae, and we all know about ATPases, which are also known as oxysomes. Now let's discuss VATPase. VATPase are present in many of the organelles of the cell, like Golgi body, lysosome. They are also present on endosomes and vesicles. Endosomes and vesicles and vacuoles. The V comes from the vesicles because these are vesicle ATPs. They are present on vesicles, and what they do is they use ATP to pump in proton. They pump proton inside the Golgi body, lysosome, endosome, vesicles from the cytoplasm by using the ATP to make a low pH environment inside these organelles. And in neurotransmitter especially, the synaptic vesicles needs proton gradient to transport neurotransmitter inside the vesicles. What it does is it first creates a proton high concentration inside the vesicle by transporting the proton inside vesicle. After making the proton gradient between the vesicles and cytoplasm, there are other transporters which can use this proton gradient to transport proton out and neurotransmitter in. So we can see the coupling of primary and secondary transport. Primary transport is done by VATPase, which uses ATP directly and convert it into ADP into inorganic phosphate, and using this ATP to make a higher concentration environment inside the vesicle, and this higher concentration is used to transport neurotransmitter inside the vacuum or vesicle. Now let's discuss AATPase. These are found in archaebacteria, not present in eukaryotes, so we are not going to discuss them. Actually, we are not going to discuss any of them other than SERCA or PAT pages. I'm just giving an overview about it. Let's see PAT pages. PAT pages, example of these are sodium, potassium ATPase and calcium ATPase like SERCA. And there are other examples of PAT pages also, but we are going to focus on SERCA. These are EAT pages. These are present on outer surface of cells, especially outer surface of cells. Example includes three main classes. One is ecto ATPase, another is CD39s, and then ecto ATPDase. They are mainly used for immunosuppressive effect, and especially when the ATP gets leaked to not hyperactivate the immune system, what we do is ATP is degraded by EATP is present on outer cell membrane of cells. This degradation of ATP causes less availability of ATP for activating P2Y receptor or P2X receptor present on inflammatory cells like macrophages and T cells, which result in suppression of immune system, especially CD39. It is present on some cells like T regulatory cells. In T regulatory cell, one of the main way the immunosuppressive effect is performed is by CD39. This cell has CD39, which can degrade the ATP and ADP both, and to AMPAS, and then this AMP is degraded to adenosine by another protein, and this degradation ensures that the activated T cells, which are already activated by the other inflammatory signs and also ATP previously, don't get activated again and again. So these are the immunosuppressive ATPases. These EATPases are also used for other factors, and they are very important in cancers and oncology. We'll discuss it when we are talking about cancer. Now let's talk about SERCA specifically. Its real structure, not the cartoonish structure that we have talked about. This is a lipid bilayer. Now let's see the peptide of SERCA. These are domains that I am making, and then this is passing the membrane, again and again. This N-terminal domain, N-terminal end, and this is C-terminus. These parts together forms actuator domain. Okay, this is the first domain that you will encounter if you are counting the amino acid from N-terminus. And then this part, let me draw it back. This part is known as P domain. or we can also call it phosphorylation domain, and this leftover part is N domain, which is nucleotide binding domain. The actuator domain can be also named A domain, and this part, this whole part of the SERCA is called as T transmembrane domain. I'm talking about these transmembrane helices and their extracellular loops. These are all considered part of T transmembrane domain, and the remaining part is known as S transmembrane domain. S transmembrane domain is kind of an accessory. It helps with some accessory function of this SERCA. They are not as important for the catalytic activity that as compared to T transmembrane domain. This is actually the diagram or the structure of all p-type ATPases. It is not specifically for SERCA, but SERCA can also be drawn by like this. So it is also part of p-type ATPase domain. P-type ATPase domain. Okay, and let me make something clear, like it is out, it is in. What do I mean by in? It is either cytoplasm. It is mainly cytoplasm. I don't need to play with it, but out one can be either extracellular fluid or lumen of vesicles and Golgi body and or other organelles. Okay. Now let's start discussing how the conformational changes occurs and how it performs its function, and what part of SERCA is responsible for what kind of function. Rather than using this complex diagram, I will just change it into somewhat cartoonish or simpler structure to understand the conformational changes that occurs and the whole process of transportation of calcium by the SERCA. So let's start with this. This is the initial conformation. Okay, in this conformation, this is P domain and this is N domain, this is A domain, and this is T and S transmembrane domain. This is the calcium binding pocket. Okay, here the two calcium comes from here. This is entry canal and binds at both sides, and when the conformational ends, then both of the calcium gets thrown out of this binding site through exit canal. But at the initial position, the only canal that is open is entry canal. Two calcium comes inside and through this entry canal and binds to both of these calcium binding sites. So first thing that occurs is calcium binds and ATP comes and binds to the nucleotide binding domain or N domain. So next diagram will be something like this is ligand calcium binding site where two calcium, let me draw calcium by red color, and after the entry and binding, even the entry canal gets collapsed by the conformational changes, and then P domain and N domain both comes in contact with each other after ATP binds, and this is A domain. There are no significant conformational changes right now in the A domain. This P domain and domain have come into contact due to the ATP binding, And as soon as ATP binds, they degrade it into ADP and phosphate. ADP remains bound. ADP remains bound at the same place where the ATP is bound, and the phosphorylation occurs for P site, for P domain. Let's draw phosphorylation by green. This is phosphate group that is added after the degradation of ATP into ADP, and this results in some changes that we are going to discuss now. And in the next step, after phosphorylation, what happens is exit gate or exit canal is open, and everything remains almost similar to the previous one. Like this is A domain, this is P domain, this is N domain, and ADP is bound here, ADP now, and there's a phosphate attached to here. This phosphate is specifically on the aspartate residue, which is aspartate 351 residue present in the P domain. And now after opening of the exit canal, and the next step is there are some changes after opening of exit canal. The conformation changes leads to decrease in affinity for the calcium in the binding site, and this results in throwing out of calcium are thrown out, but now the cell has another work to do, which is to bringing this SERCA back to the original conformation. And it is not done directly, it is done with the help of E domain, which is actuator domain. What happens is actuator domain causes opening of N and P domain from each other, away from each other. This opening result in escaping of ADP. The bound ADP gets released, and this bound ADP release is important event, as it will trigger conformational changes that will cause dephosphorylation of P domain. Previously phosphorylated domain gets dephosphorylated, and after dephosphorylation, the E domain goes back to its original conformation. Exit canal is open even now, and entry canal is closed. A domain has returned to its original position, and N domain and P domain also have returned to their initial position. Only thing that remains is these canal conformational changes that have to occur, which is next step. And now the cycle has completed, and our SERCA has gone one rough cycle of pumping out calcium outside. Either it can be from cytoplasm to lumen, or it can be from cytoplasm to extracellular fluid. Now we are going to discuss about regulation of the SERCA. As I already told, we are going to discuss about regulation of SERCA. I have drawn this SERCA structure as it is seen in crystallography. Okay, so no need to be confused about it. This is a SERCA, and this is another protein that we are going to discuss, which is phospholambens. This phospholamins is an inhibitory protein for SERCA. What it does is, let me draw it with blue pen. It can form complex and interact with SERCA, and this interaction causes SERCA to get inhibited. And how this inhibition is work? What happens is, the phospholamins, as soon as it binds with SERCA, it decreases the affinity of calcium for SERCA. No, I just misinterpreted it. I mean to say that phospholamins decreases the affinity of calcium, sorry, of SERCA for calcium. It decreases the affinity of SERCA for calcium, and this result in lesser and lesser capture of calcium by SERCA, and in the end result in lesser pumping of calcium. This pumping can be maintained or enhanced by changing the phospholamins phosphorylation state. In dephosphorylation state, it is free and can interact with SERCA freely and decrease the SERCA activity. But what if it is phosphorylated? Let's see its phosphorylated state. What happens is, it forms a complex, mainly pentamer 1 2 3 4 5. It forms a pentamer. You understand what I'm trying to say, right? Something like this. A pentamer is formed after what? After phosphorylation. When these phospholamins gets phosphorylated, they group together and become a pentameric complex, and they are now not available for interacting with SERCA and inhibiting it. So now SERCA can work freely and pump calcium out in excess amount, and this can be done by many kinases. Let's take example of beta adrenergic pathway. What happens is, norepinephrine or epinephrine can activate their adrenergic receptor, especially we are talking about beta adrenergic receptor in cardiac muscle, where this activation will lead to G stimulatory G protein activation, and alpha S subunit will activate adrenal cyclase, which will cause formation of cyclic AMP, and cyclic AMP will cause activation of protein kinase A catalytic subunit, and this protein kinase A can phosphorylate phospholamins, and this phosphorylation will cause activation of SERCA, or it will disinhibition of SERCA, which will increase the activity of SERCA, so that the calcium pumping is increased, and this calcium pumping can enhance the heart rate. As SERCA, what is the function of SERCA? It removes the calcium present in the cytosol. So after a depolarization, after the contraction, we have to remove the calcium from the cytoplasm, so that new cycle or new contraction can occur again, new depolarization can occur again. And the faster this process becomes, the faster the heart rate will become, and this faster heart rate will be accountable for norepinephrine work by sympathetic nervous system. Now let's talk about other things that influence phospholamins in SERCA. Factors which increase SERCA activity are increased SERCA activity are increased simulation of SERCA, and increased phosphorylation of phospholamin, which can be achieved by many of the mechanisms. One of them I have already told you, by protein kinase A. And factors that decreased SERCA activity, specifically SERCA 2A actually, but we are not going to in nitty-gritty details about it. These are unphosphorylated unphosphorylated phospholamine and nitric oxide can inhibit the SERCA2 activity via thiol oxidation and nitration of tyrosine residues. And another is reactive oxygen species. This concludes the regulation, roughly concludes the regulation of SERCA activity, and more of the about the 80 pages we will discuss in next video.